We actually already saw images of the next speaker in a younger version this morning. But I'm glad to say it's still recognizable. So Clark Colton was really part of the early days, and Clark has been an active participant in the biomedical community, as you know, for many, many years. And Clark also today plays a very vital role in the department, and our undergraduate major laboratory course was called 1026, which is a project course that Clark really has revitalized and put a lot of good ideas into with his usual energy, and it's become a course that many of our students really enjoy. They really feel they get a lot out of these projects, and they learn a lot. And we have a new leadership program on campus, and I was pleased to see that this leadership program has recognized what this laboratory course does for our students, much to Clark's achievement. So, and he's going to talk about education. I suspect he's Ed Merrill and the smoke sardine theory of knowledge acquisition.
Well, while we're setting up here,
your mic's not on? You need your mic.
my mic? Thank you. Well, I'm very pleased to be here today. I'm pleased to participate in this symposium to honor Ed. It has been a great influence for many of us for many years. What I intend to do is to, over the first 2/3 of my talk, to describe some current research going on in my laboratory, give you some of the flavor, and in the end I will relate that to my experience of Ed Merrill. Some of you are probably wondering what the title's about. You have to wait a little longer.
You're not on the screen yet. You need to externalize this.
We got to go to display?
Yes.
Okay. Okay.
1024 by 768 unstretched.
Unstretched? Yeah, let's go back. Unstretched. Where's that? Here, right here. Okay.
Tech displays.
You should be okay. Okay. So, I want to start off talking about two topics. And the first one is going to be very similar to what you heard from Paula in some respects. And that's enhanced imaging and therapy in cancer with targeted polymers. And the work I'll describe comes from the thesis of Kevin Brower, who's currently finishing up, as well as Michelle Miller, and some early work by Jin Zhu Dawson. The second area that I'm going to talk about is the effect of oxygen on growth and differentiation of stem cells. This reflects work being carried out by 3 students, Jeff Millman, Jit Tan, and Amanda Delano. And it was all started off by Daryl Powers, who graduated a few years ago. So, first topic is motivated by the need for improvements in diagnosis and treatment. Current imaging techniques have problems. Small primary tumors are not detected. Metastatic disease is not diagnosed. Treatment response to therapy is in fact rarely measured. And the signal in imaging often has little sensitivity and specificity for the tumor cells themselves. In therapy, the problem is that there's low specificity for tumor cells, which leads to high systemic toxicity. Now, I was diagnosed with cancer 9 years ago, and when I tell you I'm happy to be here, you know I'm telling you the truth. What I learned from my oncologist, in his view, was that we have treatments today that will do the job. The problem is either we can't see the tumor cells, or when we see them, it's too late. And therefore, recently we've put an emphasis on the imaging end of things, although as you'll see, the same approach can apply to both therapy and imaging. But the application we're looking at right now is in imaging. So we're using a polymer system involving alternating copolymer micelles. And these are created by an enzymatic polymerization with a linker, trifunctional linker, and polyethylene glycol. After the backbone polymer is synthesized, we add side chains, and then this polymer will self-assemble into a micelle on its own in aqueous solution. This chemistry was originally developed with Arthur Watterson at UMass Lowell, and we've continued to work with him to apply it to the cancer application. So the interesting thing about this polymer is it's got incredible flexibility. We can hang pendant groups of all types off the linker group. For example, we can put a fluorescent fluorophore on it. We have used side chains that are either perfluorocarbons or hydrocarbons. The perfluorocarbons we originally used because we thought we were going to do trifluorine MRI. That turns out to be technically too difficult. And so we've looked elsewhere, as I'll explain, but we've kept these perfluorocarbon side chains because it turns out they reduce the critical micelle concentration by an order of magnitude, very strong interacting, more than hydrocarbons. And we've also put on a targeting ligand combined with a PEG spacer. We use polyethylene glycol as the spacer. And the ligand we're using binds to the epidermal growth factor receptor, and it was actually developed by a student named Ben Hackle in the Dane Wittrup lab here in the department. So we have looked at, well, let me say, we can encapsulate drugs as well. But as I said, we're primarily focusing on use as a targeting agent. Now, we've looked at many parameters, the backbone molecular weight, the fraction of side chains that are devoted to targeting ligand versus side chains that do not have a ligand, the length of this spacer, and the nature of the ligand. We've actually looked at 3 so far. An interesting thing is we looked at folate, as Paul had described. That's a very common one. And after some very painful experiences, we found that with this polymer, folate folds inside. And it produces gigantic aggregates with no folate on the outside. So, as opposed to her system where the folate's displayed on the outside, because folate has significant hydrophobic character. So that's why we then went to a different ligand. So these are some in vitro uptake studies with a few polymers. This is cell-associated polymer, picograms per cell, versus incubation time. This is the size of the polymers by dynamic light scattering. You'll notice these polymers have various peaks. The conformation is probably very complicated, much more complicated than if we have no peptide on it, and if we simply have hydrocarbon alone in the interior. So what we have here is 25% side chains that bear peptide. These are all, these two are perfluorocarbon side chains. This one has no peptide. This has no side chains at all. So it's the individual chain. Nonetheless, it aggregates to form very large particles. Although, as I'll show you subsequently, we think these fall apart when they're injected into the bloodstream, whereas these probably stay intact. And what we see here is that the polymer with 25% ligands with PFC chains has very rapid uptake, much greater than the polymer without peptide. This is the one without side chains. But it does have ligand, and it has intermediate uptake. Now, we've done experiments in which we've injected these into animals and done biodistribution studies. And this is experiments done with the polymers that I just showed you. So we have the 25% PEG label with peptide, without peptide. We also have the 50% PEG with peptide, but no side chains. And then we've, for good measure, we've thrown in the peptide alone. This shows the percent injected dose per gram of tissue. It's a way of normalizing to how much you put in. And what you need to focus on here is the uptake by the tumor. And we see that with peptide, the uptake is higher than the same polymer without peptide, and substantially higher than the same polymer with peptide but with no side chains. And it's this behavior that suggests to us that the polymer with no side chains dissociates into single chains in the bloodstream. And there's a significant difference here by a factor of about 3. Our target has been a factor of about 2 because that's enough to give us something we can see selectively in PET. This is the percent injected dose in the bloodstream, and it qualitatively follows the same pattern that we saw in the tumor, except that the peptide decays more rapidly because it's a small size. So where are we headed with this? This is my last slide on this particular topic. We are going to be doing, or are doing now, these experiments may be run today, positron emission tomography. Basically, I left something out which is really important, and that is these orange asterisks are sites where we can radioiodinate the polymer. For years, people radioiodinated polymers by attaching something that contained a tyrosine group. Kevin Brower found that he could radioiodinate this polymer directly because these aromatic groups turned out to have reactivity using a standard method for radioiodination. We can also do the tyrosine and the ligand, but we don't have to. This means it's very easy to iodinate this particular polymer without any further requirements. And so just the other day, this was radioiodinated over an MGH with iodine 124. I'll tell you what that is in a moment. The iodine is very useful because iodine 125 can be used for a gamma counter. Iodine 131 can be used for radiotherapy. Iodine 124 became commercially available 2 years ago. It's a positron emitter, antimatter. And so when it is annihilated, it gives off gamma rays and it can be picked up. It's very expensive today, but maybe someday it'll be used. And it turns out, as expensive as it is, it's still a small part of the total clinical cost to do a PET scan. So it's a real possibility. So this is what we hope to get. This is actually half a mouse. This is with a passive uptake of a particular polymer that was synthesized by our collaborator, Mikhail Papasov, and it's undergoing phase 1 trials now. It operates just by the EPR technique that Paula mentioned, and so we hope to do better using the targeting. And this is a mouse. You see the material that's in the bloodstream in the liver. It also is secreted by the kidney. So you see a little bit of signal there, and it was picked up by the tumor. So we're hoping to get something like that, which will be the first time that a targeted agent has been used with positron emission tomography. All right, let me switch gears. I'm going to go to stem cells. I want to give you a little background on regenerative medicine, so we're all on the same page. How do you make a stem cell? Well, you take the sperm and the egg and you fertilize it, and it starts to grow into an embryo. And originally, that embryo consists of these blue cells, which eventually become the placenta. And in the center are the cells that will grow up to be an organism. And at this stage, they are pluripotent, which means they can differentiate into anything in the body. So what's done is to take those out and culture them. And in culture, they will self-renew. That is, they just continue to divide. And then with the appropriate agents, you can make them proliferate. We know there are ethical problems, so people have looked at other methods. One that gained some interest but still is not considered totally approvable is somatic cell nuclear transfer. Here you take the egg from one organism, and then you remove the nucleus. You take the nucleus from another organism, put it in here, then reimplant it back into the original species. For example, you can take a sheep cell, put it in here from a cow, put it back into a cow, and you'll get a sheep. So that's the second method. Still not good enough. A lot of interest has occurred over the last 2-3 years. People have found you can reprogram cells. I can take a liver cell, inject the right genes into it, and turn it into a beta cell of a pancreas. This is really very exciting. Doug Melton over at Harvard did it first. The problem is it's gene therapy. We don't know any way to do that in vitro with one gene, let alone the 3 that he needed. The second technique is to do it in a person, in vitro. And here you take the cells and you convert it back to embryonic stem cell by putting in the appropriate genes that cause it to become pluripotent and to self-renew. And that produces what's called induced pluripotent stem cells. They have similar properties to embryonic stem cells. We still don't know if they're exactly the same. They're probably not. And unfortunately, every time you make one of these, it's got its own particular set of rules, which is a problem in the field. Nonetheless, with the appropriate agents added to either ESLs or iPSLs, we can differentiate. And so this is a matter of putting the right agents in at the right time and then collecting them at the end. And I get different kinds of cells shown by the color here. But there's also a problem, because if I have any pluripotent cells left from the original stem cells, if those are injected into an animal, they have the risk of growing. And when they grow uncontrollably, you get what's called a teratoma. Teratoma is simply differentiated cells from different organs, different tissues. And it has no function. It's a tumor. It can be benign or malignant, and the FDA currently will not allow you to inject a preparation into a human that has one stem cell. So it's a very serious problem in the field, one that not many people are looking at. The next area is tissue engineering. There are people who inject these cells directly, or you can put them in some kind of construct. You do it either because they grow better, or you want to protect it from the immune system, or you want to keep these teratoma cells, these pluripotent cells inside. So if they do make a teratoma, they don't get out. And then these are eventually implanted in the appropriate place. Over the years, I've worked in the tissue engineering field. Right now, I'm doing some work on both of these topics, and I'm going to talk about both, the effect of oxygen on differentiation and the effect of oxygen on teratoma formation. Big problem. We're interested in oxygen. We spent a lot of time in the beginning playing with polystyrene cells. Nothing ever worked. And the reason is you can't control the oxygen. You don't know what it is, even if you make the right measurements. There are gradients in the tissue culture dish. This is beautiful 1032 undergraduate transport. Very simple. It's been the subject of numerous quiz problems. So you can calculate what that gradient, what the surface PO2 is that the cells see, PO2 cell. It's a function of the cell surface density, which determines the oxygen consumption rate that's enforced at the boundary. And it varies with the height of the culture medium. And you can see if I culture at 142, which is the standard condition that people use, it can be very different. And as cells grow, this is going to change. You never know what you have. I neglected to tell you why it's important. The embryo grows and differentiates anywhere from 0 to 30 millimeters of mercury oxygen partial pressure. 95% of the world studies stem cells, with 5% CO2, 95% air, which is 140, humidified, that's 142 millimeters of mercury. There's a disconnect here. God made us hypoxic for some reason. So we were interested in seeing what it is at the low end. But we faced the problem that we couldn't control it. So we came up with the idea of culturing on silicone rubber. We put a silicone rubber layer here, and what theory tells us is this is what we're going to get with silicone rubber. This is at the low, these curves. So we're insensitive to the oxygen consumption rate. We're insensitive to the height. We don't have to worry about it. And now I'm going to give you a tiny demo because Ed Merrill always gave demos. So this is an Ed Merrill demo. All right. What I have here, this is really simple. I have here a 6-well culture plate. So we take it apart. We cut off the base. Take a sheet of silicone rubber. That's, as you get it, this is protected. I've got a little sheet in here. They're stickier than saran wrap. Place it on the back, or you glue it, or use silicone rubber adhesive. Trim it, and now you've got a dish with silicone rubber on the bottom that you can control the PO2 that the cells see. Here's my Merrill demo. So anyway, we embarked on our study. I got to move along. We do differentiation. We take 500 cells, put them in a drop, let them aggregate for 2 years. They form what's called an embryoid body. Transfer them to a dish. After 3 days, change it to serum-free medium, and then we see what we get. We started out because we read the literature, and it said if you don't add anything besides serum, you get neurons. And so we were all set with reagents to check neurons. First time it was tried, student comes in my office saying, they're all beating. We had gotten cardiomyocytes, not neurons, because we were doing it at low PO2. And it turns out at low PO2, cardiomyocytes are favored, which makes sense because the very first cell that's terminally differentiated in the embryo is the cardiomyocyte that goes to make the heart. And once that's made, the next ones are the endothelial cells, because you can't get very far without creating a circulation. So it makes sense that that is where the cells would go. If you keep PO2 constant, you get 30%. We found if you vary it, you can get a lot more. You can get up to 57% cardiomyocytes, which is pretty amazing. You can use a whole variety of cytokines and get 50%, 7%. In fact, as time has moved on, people are now getting much higher than that. But here we've done it. In this case, we added a little bit of ascorbic acid. You get almost the same thing without it. There's a synergistic effect. So the main point here is that lower oxygen, it favors cardiomyocytes if you do nothing. If you do something, it can go elsewhere. So magic ingredient A will make neurons. B will send you to endoderm, which will get you into insulin-producing cells. And what oxygen does, it turns out, is that it accentuates it. And the few studies have been done, one with a colleague we have now in Portugal, you do it at lower PO2, wherever you're headed, it's better. So that's the story on differentiation. Oh, wait a minute. I do have slides dealing with the mechanism. And basically what we found is it operates later in the differentiation process. Brachyury is a marker for mesoderm, which is one of 3 lineages. The first, these are transcription factors that control the expression of the genes. The first transcription factor that you get a significant increase in is called MESP1, and work is now coming out saying that that is the key switch that channels cells to become the cardiovascular lineage. So it agrees with our work. Later on, after about 8 days, we begin to see increases at low PO2 of markers for cardiomyocytes. So the story seems to make sense. Second issue, teratoma formation. If we take the cells that were differentiated for 10 days, and now we're using reporter cells. They have in them the ability to make green fluorescent protein when they make a gene called OCT4, or this is a transcription factor. It's associated with embryonic stem cells. It's associated with pluripotency. So we can determine when cells are pluripotent by looking for green. We take it, we culture for 10 to 80 days, and then we assess either in vitro with flow cytometry, microscopy, or by injecting into nude mice to see if we get tumors. So this is what happens. If you culture these at low PO2, at normal PO2, nothing happens. The OCT4 stays the same. If you culture them at lower PO2, you get successively greater reductions in OCT4. In other words, at lower PO2s, the remaining stem cells have a greater tendency to differentiate and disappear, which is what you want. The green here shows the presence of the OCT4 containing cells at high PO2, none at low PO2. So, does this work in animals? This is what a teratoma looks like. It's a mixture of cells that look like various kinds of differentiated tissues. If you, nothing, original stem cell, in 14 days you get all have tumors. 142, it extends it by 3 or 4 days. You culture at low PO2, you now get an extension out to about 31 days. So we see low PO2, this is, I think this is for 20 days culture. It's not the full 80 days. So definitely in effect. We said, well, you know, people have suggested other techniques. Another one is to take markers for stem cells, to get an antibody to them with a fluorophore, do a FACS analysis, sort them, throw away the ones that have the marker for the stem cell, and keep the good ones. So we decided to combine that with low PO2. So we go through our differentiation, culture at 20 days, then put in an antibody to a surface marker SSEA1, which is one of the very few surface markers that are known for stem cells. This is bound to a fluorophore. Then we sort with FACS, get the positive and the negative cells. Negative means it's got none. So what happens? Well, just in vitro, the sorting, we can see you do nothing, just low PO2, you get a reduction. If you further sort, these are the cells that are positive, these are the cells that are negative. So we are getting a synergistic effect. Put them in the animal. 142, positive for SSEA, about 20 days before they all get tumors. If they're negative, we go out to 30 days. Culture at 7 days, but with the positive marker, we're out to 42 days. Do both, 7 millimeter culture and the FACS sorting. 70 days, it's actually, as of today, it's about 91 days. So this is very good evidence that, and what that means is that we managed to remove every single stem cell, or at least there aren't 2, because there are papers in the literature that have gone down to 2 stem cells and have shown that's enough to create teratomas. So we're kind of excited about this because it may be one way to treat stem cells so they can be used in humans. All right, let me use the remaining time just to talk a little bit about Ed. And I'm sure you're wondering, what does this have to do with Ed Merrill? Or does it provide insight into Ed Merrill's enormous impact on bioengineering, his methods of training students who went on to invent the field, his effect on the career of individual students, or even what was it like back then when you were a student? Because we are now in the heavy part of the seminar where we have 3 consecutive PhD students over the period 65 to 75. So, and this is when Ed was in the peak of his career. So what was it like? Well, it would be very tempting, of course, for you to say, well, look, you told us about this polymer stuff. You worked on polymers for Ed. I don't think so. I didn't. In fact, I worked on the artificial kidney mass transfer. And our conversations during the first year used to go like this. After I was, my second year, about three months, he'd call me in and say, so what have you been doing? I'd say, I've been in the library reading, which I was. So what do you think you want to work on? Mass transfer in the artificial kidney. Can I get you to work on polymers? No. I want to work on mass transfer in the artificial kidney. Okay. Another 3 months, he calls me in. What have you been doing? I won't go through it, but this happened about 3 times. And I didn't know you're supposed to work on something the professor wanted you to work on. I just got fascinated by mass transfer and artificial kidneys. So that's kind of what I worked on. But nonetheless, I nevertheless picked up valuable stuff. How did that happen? Well, this is where we get into the title of the talk, because Ed really used multiple techniques in educating students. And the assumption of this theory is we learn best when we don't know we are learning. A corollary is learn best from our environment. What was the environment? Well, number one, Ed had a lot of students. He had more students than anybody in the department. And in those days, you didn't have a limit of 2 students per faculty. So it was booming. How did he do this? Well, he had a lot of support. There were rumors at the time that he had more money than God. I'm here to tell you right now, as I stand here, that's not true. He was a close second. So, and in fact, it wasn't until I became an assistant professor that I had any idea you had to worry about money. Because anything I wanted, I got. Didn't matter. I decided I want to count tritium and carbon-14. All right, just pick out a gamma counter. You know, it was an amazing time. It was the golden era of research in this department. So he had a lot of students. One of the results of having a lot of students is they got out of control. Okay, so despite the fact that we used to give a seminar to them every year, I gave 3 instead of the 1 that happens now. He decided he was going to have his own group seminar. So once a week, we all got together, and students would get up and talk about what's going on in the lab and what are the problems and what's happening. The reason he did it, actually, was to find out what was happening, to make sure he could be on top of this gigantic group of students. But to us, even though we preferred to be in the lab and taking data, it was an enforced hour where we'd sit there and consciously or unconsciously things went in. And so as a result, I got exposed to all the polymer work that was going on just because I was forced to sit in these seminars. And I will tell you, it may have been the 1980s when Ed started putting PEG on surfaces, but PEG was one of his favorite polymers way back. And I heard about it in the 60s. So, I got to tell you, when I saw this molecule, something went off in the back of my mind, and a little voice said, Ed Merrill would love that molecule. Because as Ed said, the PEG is waving in the breeze. It's like seaweed sloshing back and forth with the tide, and it's going to prevent the immune system from recognizing it. So I knew that immediately. I don't know how I knew it, other than the fact that I knew what Ed was thinking. I knew what his students were working on. And so those seminars actually were very valuable. And I only found out how valuable they were later when I discovered I actually knew something about polymers. So let's get back to the theory, because we still have the whole silicone rubber thing. And where did that start? Well, the hypothesis of this is that the very best learning is what we learn from each other. The more we interact, the more we learn. Therefore, the closer we are, the more we learn. And so this theory suggests that the efficiency of knowledge acquisition is proportional to 1 over AS. AS is the specific area, feet square per student. I did my thesis in the penthouse. Yes, see? But you're one of the few people who knew. Not everybody knew about the penthouse. The penthouse was kind of secret. I later figured out that this is where Ed Merrill put his students who were not working on polymers. But at that time, I viewed it Well, in retrospect, I now view it more like Skull and Bones. You know, secret society, nobody knows anything about it, but they all did very well. So, how did this work out? Well, as I said, it was called the penthouse, but it really was a secret room. Not everyone knew about it. I don't know if any of the, I had several thesis advisors. I'm not sure any one of them ever came to visit me. It just wasn't on your regular itinerary, and it was tough to find. So, just to give you an idea what this is all about, I took my iPhone 3G 2 megapixel, low resolution, around the Institute to try to see if I could reestablish the penthouse, okay? And it's there are only a few windows in the whole Institute where you can get a view of the penthouse. So this is building 12. As you can see, the fact that it's called a penthouse, you know, you think you got a great view, but it was a one-story building. So, anyway, this, after a lot of soul searching, I've decided that the penthouse is right in here. You can see it's got some windows here. It's got similar windows on the other side. That puts it right here. On the other side of these smokestacks are air handling things. They were here and they were air handling everywhere. So, I entered Building 12. Building 12 today is Career Services, if you can read this. And they interestingly, they had a floor map. Not many buildings that might have a floor map. So I looked, I'm over here, and I'm trying to find, there's no second floor. And then I spotted a stairway. It's got down and up and nothing else, right? And it's next to room 144A. So presumably, if this was a real room, it would be 244A, okay? So I went, and this is 145 here. I went over to 145. It's 2 o'clock in the afternoon. It's locked, and it's dark. So immediately I was suspicious. And I looked around for someone who could get me into the, quote, penthouse. Zero. Nothing. Then I found some people. Then I explained, there's a stairway in there, and I want to go up the stairway. Why? So I found someone, secretary, that I basically said, look, I'm a professor here, you see? Just taking some pictures. Will you kindly open the door and let me in? So she let me in. And lo and behold, I went to opened the door and I found the spiral staircase. I used to think I was on a submarine. So here we're going. This is the top of the submarine, the spiral staircase. If you look to the right, it was all air handling. And if you look to the left, you saw the secret room. Okay? And you don't believe me, it's secret. This is the only room at the Institute that doesn't have a number on the door. So I go inside and I feel at home. Why? These windows, which have not been washed since 1921, are exactly the way they looked when I worked there. There was a wall where there were some tables and chairs. That were 2 desks. Here is a bench. That's a bench that was here then. It's got some drawers. You can see a couple of outlets. No water on this side. There was a bench, one other bench, and it had a water faucet. We had a table in the middle, like, you know, that big. Multiple people were working in there. And then I found the guts of the room was actually a cage that you could lock. And that was used either to store boxes or people. I'm not sure which. So, this was an incredibly productive place. Who was up in the penthouse when I was there? Well, and when I showed up, it had David Graves. He became a professor at Penn. Is David here? Ah, there you are. Okay. Well, you're going to relate to this. Richard Buckles, research director of ALZA. Then I showed up, and when I showed up, there was no room for me. There was no table and no chair. There was a bench where I could set up equipment, but that was it. It's one reason I spent so much time in the library. So then I think David left. John Day came up. He's now president and CEO of Mining Services International in Utah. Peter Strove did his master's thesis and I think stayed in the same room for his PhD. He's professor and was chairman at UC Davis. Peter Farrell chanced in to do a master's thesis. on membranes with my equipment. Became professor of biomedical engineering at University of New South Wales, Australia. 20 years ago, founded and became chairman of ResMed, a $5 billion company listed on the New York Stock Exchange. And today he's a major donor at MIT, but even more so Harvard. So these are the people who were related to Ed Merrill, master's theses, PhD theses. There were also some hangers-on who came by all the time because they were friends or we were doing something. One is Doug Cortez, who became president of Florida Development Corporation. He's now retired. Doug was friends with John and I. We formed a investment club. We got money from our parents, pooled it. We had $5,000. We went downtown, Hornblower and Weeks. We found a stockbroker who made it disappear. He managed to do $165,000 of trades in 6 months. And that was real education, let me tell you. But we were busy, you know, we didn't have to. Then there was H. Kent Bowman. Kent was together with Doug, John, and I, actually formed a company. We called it Technology Recruiting. The basis for the company was the following. If you smelled like you came from MIT, you got a job offer. I had 11, before I decided that I was going to be on the faculty, 11 job interviews, 11 plant trips, 11 offers. It was incredible. So we said, we got to capitalize on this. So we decided to write a book on how to recruit MIT students. Well, it had a not great ending. Nonetheless, we didn't suffer. I'll tell you in private about the ending if you want to hear about it. But anyway, Ken Bowen became a professor down the street at materials science for 20 years, and then he decided he went to Harvard Business, and he's now a professor at Harvard Business. So you can see it was a fairly distinguished group up there. They did not suffer because of the effect. I think they prospered. And so I've done actually some calculations here on the productivity of the penthouse, all right? It's 15 by 15 feet square. I walked it off with my shoes. 240 square feet, slightly larger than my current office. There were 2 desks, 2 chairs, some cabinets, some bench space, 1 refrigerator. On average, 4 occupants. So it was 60 square feet per student for our specific area. The current MIT benchmark, whatever that is 280. Our department, which is filled to the gills, is 140. So we were tight by anybody's standards. So then I came up with another productivity index, which is, this is a proposed productivity index, PPI. It's the area per publication. And I had my 60 square feet, 10 papers ultimately came out of my thesis. That's 6 square feet per paper. And I am willing to bet that Ed Merrill and his academic descendants are extremely high in this parameter, which will become a new benchmark for rating graduate programs. So I would like to thank Ed for the environment that he created that allowed me to prosper and so many other people who are associated with him to prosper. And the success of his students, I think, ultimately is related to the smoke sardine theory of knowledge acquisition. Thank you.
Well, I wish to thank Clark for having solved our space problems. I'm sure that Paula would love to use that index in the next discussion with anyone that needs space. I think the sardine index sounds better. But if there's anyone that has a real brief question.
I'll try and make it brief, but I'll tell you how Clark and I learned about gas chromatography. This room 244 was right over the faculty offices. And since Dick Buckles is not here to comment, I'll just say...
I need to make a comment about him, yes.
But anyway, we'll blame it on Dick. Occasionally there would be a small spill of solvent. we quickly learned that the air conditioning system had a hold-up time of about 20 minutes. So if we spilled a solvent, we would either get out of there very quickly, or we would get busy on a piece of paper having nothing to do with solvents. Because somebody, I think it was usually Brian, would come charging up the stairs saying, "Who's using solvents?" And of course, there was no evidence.
I'd just like to make a comment about Richard Buckles. Richard was one of the people who came from Cornell. There was a whole batch in the 60s of really good people, and he was very bright, very smooth. He had a similar pedigree as Sam Bodman, who preceded him by a year or two. Both top of their class at Cornell, both active in student politics. The only difference was Sam was treasurer, Buckles was president because he was better looking. So, just one comment. The silicone rubber, I forgot to tell you. I learned about silicone rubber from Dick Buckles. Silicone rubber was one of Ed Merrill's favorite polymers besides PEG. And it had just been recently invented, and they'd made two capillaries out of it. And so Buckles was studying blood oxygenation in silicone rubber capillaries, doing the modeling. I sat next to him. I knew everything he did. So one day, after he left, I get a call from Charles Satterfield saying, "I consult for Monsanto something or other over in Medford. They got a problem. They got a government contract to build a fuel cell, and they're not trying to figure out how to get oxygen out of blood. Can you help them?" I said, "Sure." I go over there. We decided I was going to do a model of deoxygenation of blood. So I just took Buckles' model, turned it around backwards, made some changes, and then got the results. And I was paid 50% of my annual MIT salary. So at that point, I learned that consulting can pay off. So we wrote a paper, and I put some diffusivity and solubility parameters in the paper, just pulled them right out of his thesis, and I used the old trick. I said, "This is what we use for a calculation." I didn't have time to go check them, so I just said, "Want to record what we used." Over the years, I got back into silicone rubber because I knew about it from working in Merrill's lab. And my students started to make models.
Maybe you can finish.
20 seconds.
All right.
My students are making calculations with silicone rubber. We're starting to publish this. I remember, I have no idea if this is right. So I said to Jeff Milliman, "You must go back to the literature 50 years ago and tell me if these numbers are right, because I have no idea if they are." So he went back and he came back and reported, "We're good." So they were correct.